Hello everyone, I am again Qazi Fariduddin representing the Department of Botany, Aligarh Muslim University, Aligarh. I extend again a warm welcome to all of you for this plant physiology and metabolism course. In this lecture, we are going to study the most important unit of plant physiology, namely photosynthesis. And over a period of time, we will dive in fascinating world of photosynthesis. Here is a brief overview on the contents that we are going to discuss today and in upcoming lectures. So as we know that the ultimate source of energy on the earth is sun, and photosynthesis is most crucial biological process which is capable of harnessing this light energy and converts it into the chemical energy. The literal meaning of photosynthesis is basically what? Synthesis with the help of light, where what happens, light energy is converted into chemical energy and which is stored in the organic matter in the form of carbohydrate. So this carbohydrate along with oxygen form the end product of photosynthesis. I will take you back little bit in past. Julius Sachs in the year 1862 showed that photosynthesis occurs in chloroplast and results in the synthesis of starch. Even though the main framework of photosynthesis was finished by the middle of the 19th century, it took almost another century for the structural and chemical specifics of photosynthesis that was revealed by modern techniques of microscopic and radiochemical study. Photosynthesis is a physiological process. Just I mentioned that where plant use light energy to synthesize food material, and I would like to share a very interesting fact that of the total photosynthesis, about 90% is carried out by algae. 90% is carried by marine algae, and the ratio of the volume of the O2 released in the photosynthesis and oxygen utilized in the respiration during the day is about in the ratio of 10:1. And basically if we talk about what is the nature of the photosynthesis, so it's an oxidation reduction process where water is oxidized and carbon dioxide is reduced to glucose that is stored in, that is a form of carbohydrate as depicted in this figure. So in higher plant, generally oxygen comes from water, not from carbon dioxide. It was earlier myth that oxygen comes from carbon dioxide. So, and if oxygen is involved in the process of photosynthesis, it is called oxygenic photosynthesis, that is commonly found in eukaryotes and cyanobacteria. And if oxygen is not involved, it is called anoxygenic photosynthesis, that is generally found in photosynthetic bacteria. But I would like to share you another interesting fact. For evolution of one molecule of oxygen, 4 water molecules split. So a total of 6 oxygen molecules evolve, hence it requires 24 water molecules to split. So now what is the basic organization of the photosynthetic apparatus? Just it is very well evident in this figure. Generally the most active photosynthetic tissue in higher plants is mesophyll of the leaves. So this is the site where chloroplasts are present, and in these chloroplasts photosynthesis takes place. Chloroplasts of higher plants are generally discoid or ellipsoidal in shape, measuring about 4 to 6 micrometer in length and 1 to 2 micrometer in thickness. And these chloroplasts are surrounded by two membranes, outer membrane which is evident in this figure and which is relatively freely permeable, while the inner membrane is rather selective permeable. So the space inside the chloroplast is generally divided in two compartments, namely one enclosed within the thylakoids called lumen and the other outside the thylakoid, which is called stroma. So basically thylakoids are what? Thylakoids are sac-like structures, as you see in this figure, where chlorophyll is present. Hence, these thylakoids serves as a site for the light reaction of photosynthesis. Carbon reduction reactions, on the other hand, are facilitated by water-soluble enzymes, so they take place in the stroma of the chloroplast, as stroma is hydrophilic in nature. Further, the thylakoids are closely associated and form stacked membranes called grana lamellae, while exposed membranes without stacking are known as stroma lamellae. So you must understand the difference between grana lamellae and stroma lamellae. And generally, each chloroplast consists of about 10 to 100 grana, and both these thylakoid membranes and stroma lamellae are made up of lipid bilayer and protein as usual. Apart from the enzymes required during light and dark reaction of photosynthesis, some ribosomes and DNA have also been found in chloroplast. That's why it is referred to as semi-autonomous organelle. So this is the figure. You can see that all the membranes what I just mentioned and lamellae, basically this is the internal structure of the chloroplast from the Pisum sativum, and this was revealed by transmission electron microscope. Now next aspect, how solar energy is captured by plant. So solar energy or light energy actually received by plant, and in this order light must be absorbed, and of course there are certain light absorbing molecules, and these complexes are called photosynthetic pigments. These pigments are found in the thylakoid membranes of the chloroplast, which occur as complexes bound with proteins. Basically, chromophore is the main light absorbing component present in these pigment molecules. Now, on the basis of structure of this chromophore, photosynthetic pigments could be classified as chlorophylls. And now question is, what are the chlorophylls? So these are the basically principal photoreceptors in the plants, and it consists of cyclic tetrapyral ring termed as porphyrins, and which serves as a head. So the structure of these porphyrin resembles that of hemoglobin in the animals. However, in hemoglobin, iron is present in center of this porphyrin ring-like structure, but in chlorophylls, magnesium occupies the central position. The porphyrin ring contains 4 pyral rings which are joined together by carbon-hydrogen bridges, as you see in the figure. Also, a long chain of carbon atoms is attached to this porphyrin ring, and this is called phytol chain, which is generally made up of hydrocarbons. This phytol chain allows chlorophyll to be soluble in organic solvents. In higher plants and green algae include two main types of chlorophyll, chlorophyll A and chlorophyll B, which are often found in a ratio of 3:1. Chlorophyll is basically universally present in all organisms that carry out photosynthesis, whereas chlorophyll B is present in higher plants and green algae. Also, two chlorophylls differ because in chlorophyll B, an aldehyde group is present instead of methyl group present at the third carbon atom in second pyral ring, as you see in the figure. Chlorophyll C is present in diatoms, dinoflagellates and brown algae. Chlorophyll D in some red algae and chlorophyll E in zoospores of Vaucaria. Now, one general aspect I would like to share that chlorophylls absorb light most strongly in the blue and red regions of electromagnetic spectrum, but poorly in the green portion. So this is the main reason why leaves appear green in color. Now, in addition to chlorophyll, there are certain other major light absorbing molecules. There are two groups of accessory pigments which absorb light in the wavelength region where chlorophyll do not absorb strongly. So this is the difference. So these are the carotenoids and phycobilins. Now, carotenoids are basically found in all natural photosynthetic organisms, and they absorb blue and green wavelengths and therefore give plant a yellow or orange color. They consist of open chain conjugated hydrocarbons which end on both sides by ion ring, like for example in lycopene, which is a chief pigment in tomato fruit. Its ring in both ends are open and not closed. These are the carotenoids. These carotenoids are basically distinguished from one another by their presence of end groups. So their general formula is C40H56. The light absorbed by the carotenoids is generally transferred to chlorophyll to carry out photosynthesis. Because of this role, they are called accessory pigments. So they are further subdivided into two classes. Xanthophylls contain oxygen and carotenes without oxygen. So you might have observed that during autumn, just before when leaves fall, chloroplasts are degraded, and now these carotenoids pigments, which are comparatively more stable pigments, are unmasked and they now impart yellow or orange color to the foliage during autumn season. That's why plants generally impart different colors in autumn season and give a very beautiful scene in the plants growing in temperate region. So the role of carotenoids along with the other compounds like flavonoids during color change in autumn leaves has been illustrated in the figure. So these carotenoids play an important role in photoprotection of chlorophyll against oxygen radicals. Now, in addition to this, there are secondary accessory pigments, namely phycobilins, and these are red and blue pigments. So it serves as accessory pigments. They are also trap light energy and feed it to the chlorophyll molecules present at the photochemical reaction center, and they are open chain linear tetrapyrrole that lack magnesium and phytol chain. These photosynthetic phycobilins are phycoerythrobilin, phycocyanobilin and L-phycocyanobilin, and they are covalently bound to water soluble proteins to form phycobiloproteins. There are two major phycobiloproteins, phycoerythrin and phycocyanin. Now these phycobiloproteins aggregate into cluster that adhere to the membrane called phycobilosomes. Phycobilosomes are present abundantly in red algae and cyanobacteria. So how light energy utilized by photosynthetic pigments? So whatever the light energy falling on the surface of the leaves is not absorbed by pigments, but some of it is reflected and some is transmitted through them, and only you see that a little portion of it, about say for 1%, is actually absorbed by these pigments. So photosynthetic pigments absorb light energy only in the visible part of the spectrum, and that ranges between 400 to 700 nanometer. And these radiations are photosynthetically active radiation. So there is a very weak absorption by pigments in green part of the spectrum, so it is reflected mostly, and hence chloroplasts appear in green in color. Now, what is absorption spectra of photosynthetic pigments? So, basically this represents the range of light wavelength that cannot absorb during photosynthesis, and chlorophyll exhibits peak in the blue region around 430 to 400 nanometer and red around 640 to 680 nanometer regions of the spectrum. Therefore, it can be said that basically they chiefly absorb light in violet blue region and red part of the spectrum. Other pigments such as carotenoids broaden the absorption spectrum by capturing light in different regions, particularly in the blue and green wavelengths. So, peak of different photosynthetic pigments have been illustrated with different colors in this figure, and you see the difference in absorption spectra of different pigments. Now, what is action spectra of action spectra? So, as you see in this graph, action spectra is the rate of photosynthesis at different wavelengths of light. So, the action spectrum of photosynthesis is obtained by plotting a graph between the rate of oxygen evolution by a plant under different wavelengths of light. So, the first action spectra of photosynthesis was given by Engelmann. He used a spectrum of visible light that was allowed to fall on oxygen seeking bacteria along with green algae. Now, what happened that bacteria accumulated in the regions of algae exposed to blue and red light, which indicated maximum evolution of oxygen that occurred in blue and red region of the light spectrum. And further, this indicated that the rate of photosynthesis being maximum in blue and red light. Also, as we discussed in the absorption spectra, that chlorophyll absorb maximum in blue and red region of the light spectrum, which indicates the role of chlorophyll in carrying out photosynthesis. Besides determining the connection between the chlorophyll pigments and the photosynthetic process, studying the action spectrum has been essential in understanding the photosynthetic light reaction and supposed to be very important for discovery of, supposed to be served as a very important role for the discovery of two distinct photosystems operating in evolution of oxygen during photosynthesis. Now, I would like to share the story how two photosynthetic systems were discovered. So, the discovery of Robert Emerson and C. Lewis in the year 1943 explained the action spectrum for photosynthesis in the visible region of the light spectrum, while they performed the experiment using the green algae chlorella. And in this experiment, what they did, they determined the quantum yield of photosynthesis in this algae. Quantum yield is what? Now, question arises, what is quantum yield? Quantum yield is the number of oxygen molecule released per photon of light absorbed. As absorption of 8 photon yields 1 oxygen, they assumed that quantum yield of 0.1 should be fairly constant for light absorbed by chlorophyll molecule. In the experiment, they reported that value of 0.1 quantum yield was notably constant over most of the spectrum. However, a sudden and sharp decrease in the quantum yield took place at wavelength greater than 680 nanometer. Even though chlorophyll still absorbs photons in that region, so this drop in quantum yield in the red region of the spectrum was termed as red drop phenomena. Hence, this experiment concluded that far red light, that is more than 680 nanometer, alone was not sufficient in driving photosynthesis. Later, Emerson and co-worker in the year 1960 revealed that when the inefficient far red light, that is beyond 700 nanometer, if supplemented with light of shorter wavelength, that is red light, that is 650 to 630 nanometer, the rate of photosynthesis was 2 to 3 times greater than the sum of the rates obtained with each beam of separately, and this was termed as Emerson-enhancement effect. Thus, it now became clear that for photosynthesis to operate with two efficiency, both wavelengths, that is 680 nanometer and 7 nanometer, must function concurrently. And this experiment concluded the existence of two pigments in photosynthesis, that is photosynthesis 1 and photosynthesis 2, while photosystem first is primarily activated by far red light, photosystem second is activated by red light. So, what is the fate of light energy absorbed by photosynthetic pigments? Just a basic I would try to reveal. So, a quantum of light energy is generally represented by what? That is a photon. So, the normal state of any molecule or atom is called as its ground state, that is could be represented as S0 or singlet state. When the electrons of this molecule, a particular molecule absorbs a photon, it then rises to a higher energy level called as excited second singlet state, that is S2, which is unstable with half-life of 10 to the power minus 12 seconds. Now, this electron comes to next higher energy level and extra energy is released in the form of heat, that is called as excited first singlet stage, that could be represented as S1. This is also comparatively unstable with a half-life of about 10 to the power minus 9 seconds. Now, from first excited state, that is S2, the excited electron returns to the ground state in two ways. One is it may lose extra energy in the form of heat. Second, it may lose extra energy in the form of radiation energy and is called luminescence. So, if the luminescence arises because of the transition of electrons from a singlet excited energy state S2 to a singlet ground state S0, then it is called fluorescence. The excited electrons may also lose its energy by internal conversion and comes to another excited state called triplet state, that is T1. So, this state is metastable with a half-life of about 10 to the power minus 3 seconds. Now, from here also the excited molecules may return to ground state in three ways. It may lose extra energy in the form of heat, it may lose extra energy in the form of radiation energy, and this is called phosphorescence, that is returning from T1 to S0 state. Next, the electrons carrying the extra energy may be expelled from the molecule and may be then consumed in further chemical reactions. So, this then what happens with the excited triplet state of chlorophyll A molecule, which takes part in light dependent reaction of the photosynthesis. Now, there is a division of labor between antenna complex and reaction center pigments. In all photosynthetic system, photosynthetic pigment molecules are bound to proteins and forming pigment protein complexes called pigment systems or photosystems. Each pigment system, that is photosystem 1 and 2, has two components. Photochemical reaction center, this is represented in the form of protein pigment complex that brings about photochemical reaction. There are two types of reaction centers, FES type reaction center present in photosystem 1 and pheophytin quinone type reaction center that is present in photosystem 2. Antenna complex, that is light harvesting complex, basically captures light energy and transfer it to the reaction center by process called resonance energy transfer, and the size varies from 200 to 300 chlorophyll molecules per reaction center in hair plants. The two photosystems are spatially separated in thylakoid membrane. PS1 is located mainly in the stacked oppressed regions of thylakoid membranes. So, at the end of the lecture, we have learned the basic outline of the photosynthesis and pigments required for successfully carrying out the photosynthesis. In my next lecture, I will dig further the detailed mechanism of light reaction. Thank you very much.